From McMaster University, I'm John Preston, and you're listening to Big Ideas for a Changing World. In this series, you'll hear from researchers from McMaster's Faculty of Engineering and beyond who are creating innovative solutions to our world's greatest challenges. I have the chance to see some of these solutions up close as the faculty's Associate Dean for Research and External Relations. With coronavirus still well in control of the world, everyone is thinking about viruses, everybody's thinking about vaccines. And one of the important things about a vaccine is getting it to the people who actually need it. So today's episode is about a new breakthrough which will enable vaccines to be delivered more effectively to many more people around the world. With that, I would like to welcome Carlos Felipe and Saher Esmaili Samani to today's recording. Let us begin by asking you to introduce yourself.
My name is Carlos Felipe. I'm a professor in the Department of Chemical Engineering. I've been with McMaster for 18 years now, and I currently also serve as the chair of the department.
Thank you for having us here. I'm Saher Esmaili Samani. I'm a research engineer in the Department of Chemical Engineering. I completed my master's program at McMaster University in the field of chemical engineering and biochemistry and joined the startup company since March 2020.
Thank you so much. So today's innovation, I've been following this for quite a while. And Carlos, back at McMaster Engineering, I would ask you periodically about how it was going. It was a long process to deliver an overnight success. Can you tell us a little bit more about this delivery mechanism for vaccines that was recently published in Scientific Reports and that also was on the front page of the Globe and Mail?
John, so you are absolutely right. It's been a long journey, and actually, coincidentally, it started when we had the last outbreak here in Canada with SARS. At that point, there was a large group of researchers here at McMaster as well as throughout Canada that we got together and got some substantial amount of funding from the federal government and CERC to develop detection technologies, virus and pathogen detection using something called bioactive paper, analogous to the pH strip, the litmus test. But instead of determining what is the pH of a solution, the goal was to detect the presence of a pathogen. That was a long time ago. As part of that project, we had to develop technologies that made that litmus test for pathogens possible. And one of the challenges that we faced was to being able to preserve and protect very delicate biomacromolecules, biological species, from degradation in the field. You can imagine having the piece of paper. If you have to use it somewhere else, you cannot keep that refrigerated. So we have to develop a technology that provided the solution for that. So that was the genesis of the solution that we created for vaccines. It was not motivated by specifically targeting vaccines, but we saw the path once we started getting some initial results on that project.
That's a fascinating history, and it kind of tells us how research actually works in many cases. So now we have a stable, affordable way to store fragile vaccines for weeks at a time. Can you explain the technology, how exactly it works?
Yeah, so can you imagine that vaccines, when you go to your favorite supermarket to get the annual flu shots, you notice two things: that the pharmacist is going to go into a refrigerator and pull a vial of the vaccine. So that tells you that whatever is inside that vial, it's fragile and needs to be stored at low temperatures. What is inside that vial is a variety of proteins, some viral particles that you can imagine that as temperature increases, these things start to melt and fall apart. When they do that, they essentially lose their ability to be effective. What we develop is a technique that we take that vial of solution, and we had 2 sugars, and essentially we dry it down. What is formed at the end is a very thin film that contains the vaccine inside it. If you imagine yourself within that film, and if you are a viral particle, what you're going to feel like is trapped and unable to move. So the viral particles cannot interact with each other, and they cannot unfold because they are totally surrounded by this wall of sugar that prevents them from changing shapes. If you want to use a vaccine, the only thing that you have to do is to add water, because that film is made of sugar. It will immediately dissolve, and the viral particles that are part of the vaccine now can float into solution, and they are ready to be administered to a patient.
That's truly amazing that you stop the vaccine or the key ingredients from melting simply by holding them in place. Is it really that simple? It sounds like stirring cream into coffee. And how would the end user actually end up taking the vaccine?
Yes, so of course, mixing sugars and vaccines is a simple thing to do, but the question is that what would be the result of that mixture? There are some questions here, like how we should mix them, what kinds of sugars we should select, and through which process steps, and what would be the final formulation of the mixture. And our technology actually answers this question because our team have developed a precise formulation for a stabilization of vaccines that can eliminate the need for vaccine refrigeration during transportation and storage by selecting specific kinds of sugars that have been approved for human consumption and for injection in the human beings, and also by designing a specific and precise process steps like how we want to combine the ingredients, how we want to dry them, and form the final product. And about how does the end user can take the vaccines, it's very simple, like any other vaccine. The end user, like clinicians, can restore the vaccine by simply dissolving it in the water and just administrate it to the patients like they normally would. So we are using Polulan and Trehalose. So like Trehalose is a disaccharide that already is being used in the market for a stabilization of the pharmaceutical products. And also we are using Polulan, which is a polysaccharide. So like combining the highly advantageous properties of these two sugars enable us to stabilize the vaccine in like such a way that they can tolerate without refrigeration, being transported in temperatures up to 40 degrees Celsius for a couple of months. So they would be stable. And about the final product, it's like a dried format that can be in a powder form, in a film form, as a coating.
Carlos, tell us more about these sugars. What is Polulan and what does it taste like?
Probably you already actually tried Polulan at least once in your life. It's the sugar that is used to make the Listerine breath strips, those strips that you buy in the supermarket and that you put in your tongue, and it rapidly dissolves and gives you this amazing breath fresh. That is made possible because Polulan, it readily dissolves, is the sugar that is utilized to make Listerine breath strips.
Carlos, the ultimate goal is to make the vaccine stable. Can you tell us a little bit more about how that happens?
Sure. So vaccines, they typically require refrigeration at 4 degrees, or in some cases actually minus 20 degrees, so that they can be moved from the production site to the point of administration. Thermal stability for our case, or our definition, is the ability to actually transport the vaccine, but now at 40 degrees and for periods as long as 4 months. If we are able to do that, we call that vaccine is now thermally stable. It's extremely unlikely, almost impossible, that a vaccine is going to be exposed to such a high temperature for such a long period of time. But if that happens, we define thermal stability by still having a viable vaccine at the end of that very harsh set of conditions.
This is going to have an amazing impact in a country like Canada, but really I can see that this would have an even greater impact in other places around the world. Can you maybe elaborate on that?
Yes, for sure. So currently, almost all available vaccines are thermally liable and must be stored at temperatures between 2 to 8 degrees Celsius all the time from production to dispensation in order to maintain their efficacy. And maintaining a refrigerated distribution system accounts for up to 80% of the cost of vaccination. Our technology eliminates almost all the cost of transporting vaccines because it does not require refrigerated transportation. And the method creates light, durable, and compact doses that would be ideal for shipping any kind of available vaccines all over the world, not only to like very developed countries like Canada and US, for sure to developing countries that extremely need vaccines, but they have limitation in their infrastructure to have the vaccine. And I can say this technology has the potential to really change the way the vaccine being delivered and administered around the world.
Carlos, so you've got a company named Elerex. Can you tell us about where the company is at? What size is it? What are its activities going on at this point?
Yes, John. So our company currently has 5 people. We are incorporated in January of this year. We got some initial runway funding from private investors. We have a CEO, we have an expert in intellectual property. We have 2 people working in the lab and 1 person that is responsible for research. That is me at this point in time. And our focus is working with a client right now to bring this technology out of the lab into a production facility and solving the problems of scale up and quality assurance, reproducibility. So that's our focus. This point is one big project that we want to learn from it as much as possible and to bring this technology to be as effective as we believe it can be.
For someone who's had a significant career in academia, can you comment a little bit of what it's like to get involved in a startup culture?
It's very different. I never thought that I would do it. When we did the research, and actually when we saw the initial results with the vaccine, actually it never crossed my mind of having a startup company. But there were several reasons that kind of changed my mind. The first one was that when the paper was published, there was a fair amount of attention that was brought up, and investors approached us, companies that were interested in the technology. So that's, I think, was probably the first step in changing my mind and actually looking into the entrepreneurial aspects of the work in a slightly different way. I think that validation by people that work in industry is a pretty powerful catalyst. The second is that in engineering, particularly in the Faculty of Engineering, there's been quite a strong focus and efforts in promoting entrepreneurship. And if opportunities like this arise, I think it is the duty of a principal investigator or a professor to actually take these opportunities to try something new and to provide an example that's okay to try. And it's hopefully things will work out. But the challenge is quite interesting. It's very unusual. The third thing is that actually I couldn't have designed a better timeline because this past year I was on a sabbatical. It was like perfect timing to try something different while being on sabbatical.
Sahar, for you, this was also a change coming much earlier in your career, but I actually hear that you joined the startup right around the time that the pandemic hit, March 2020. Can you tell us what it's been like, both in terms of getting involved in a new startup and then doing so in the reality that we face with COVID-19?
Yes, exactly. I joined the startup in March and exactly when the pandemic hit. And I can say I was shocked at that time. And I think all the people around the world were shocked at how fast the virus can spread and change the world situation. And it was a unique experience for me because my lifestyle was changed. And at the same time, I started my job. And I was working from home at that time to do some background researches and design the experiments in advance. But with the help of McMaster University guidelines, we were able to come back to the lab and start the work, which was really good.
So you've had a very interesting career path already. What advice would you have for student researchers who are interested at being at the intersection of health science and engineering, and then maybe going into innovation afterwards?
Yeah, well, I can say that it's really interesting to work in both fields of engineering and health sciences. When I see that how can I gain knowledge in one field and apply that knowledge to the other field, really excites me. And students who want to take this path also first need to gain a good knowledge in both of these fields. And they want to have the ability to make use of that knowledge in the other field to be really successful. And they just need to be careful about how people's approach for problem solving may be different in these two fields, and they should make a good connection between these two.
So what are the timelines, Carlos, for bringing this innovation to market?
Realistically, I think it's going to be a couple of years. As part of the startup, we were very fortunate already to secure a project with a major pharmaceutical company to test our formulation with a couple of vaccines that they already are in the final stages of approval and development. It will take, my estimate, it will take maybe two or 3 years. But at the same time, we are cognizant that applications of vaccines for humans, it has a lot of hurdles in terms of approvals. What we are also working on is applying this technology for animal and pet vaccination. It's a huge market. It's a multi-billion-dollar a year market. And I think that the barriers to entry or for approval are substantially easier. And I think it will probably provide additional comforts to other companies that have human vaccines to look into this technology with a little bit more of confidence.
We've heard that from a number of groups, that looking at multiple ways to bring their innovation to markets, taking account of different timescales. Of course, the issue that people are most concerned with right now is a vaccine for COVID-19. I suspect you've been following the various vaccines that are under approval. One of the issues that I've noted is that although some people are optimistic about the first vaccines coming out maybe as early as the fall, if you look at the number of doses that would be created, having the vaccine available for everyone worldwide is going to be a multi-year process. Will your approach be able to work with these vaccines that are under development? Are there restrictions of types of vaccines that it would work for and others that it will not?
Up to now, we have been pretty fortunate that we have been seeing positive results with a variety of vaccines, a variety of enzymes, antibodies, RNA, DNA. So quite a versatile technology. So although we didn't try yet for the specific COVID vaccine because it's just not there yet, pretty confident that we can adjust the technology to make it possible. And I think it's going to be, perhaps might play an important role in terms of stocking or building stocks of vaccines that people will probably, or companies will produce large amounts, and there will be a time delay between production and distribution. And I think that this preservation technology might help alleviate some of those problems as well.
This technology was, I presume, not developed in isolation in your group. Can you tell us about the collaborations that you needed within the university to bring this forward?
As I've mentioned, this was a pretty long road and we had to do multiple stops along the way. Every time that we stopped, we invited somebody to join the trip to make it successful. We started with some people in the Department of Chemical Engineering. Bob Pelton was the creator of this research network. And we work very closely also with John Brennan from chemistry and Ying Fu from biochemistry. But when we started working with the vaccines, we reached out to colleagues in the Faculty of Health Science, Ali Ashkar and Matthew Miller. And these guys are rock stars in that area. It's pretty amazing that at McMaster just took an email, and a couple of days after, we already doing the projects together. And if you look at the paper from Scientific Reports, you will see a large collection of names from people from various departments. So it took an entire group of people to make that technology possible.
That's something that we're hearing on in a regular sense in these podcasts, that McMaster, one, is a great place to have collaborations, and two, that collaborations are absolutely essential to bring great ideas all the way through to application. Carlos, you were known on campus for your sweet tooth. Essentially, you spend most of the time roaming the halls looking for candy, often sipping on a Diet Coke. And I'd never understood that. Like, why Diet Coke if you're eating candies all the time? How are you getting by in the pandemic without your usual sugar sources?
I have my usual sugar sources. I just have to go to the supermarket and buy them. So I have an ample stock of Coke Zero at home, so I'm okay. And candy. And it's ironic that actually the technology that we developed to preserve vaccines actually uses sugar. So I knew that I was on the right track of always looking for sugar, as in the last 12 or 15 years that I've been in the Department of Chemical Engineering.
Just goofing around, but we may end up using that. Saher, what's next for LRX, and what will the team be doing in the weeks and months to come?
Yes, so what we are doing currently is that we want to adapt our technology for other commercially available vaccines. So until now, we demonstrate that this technology is working very well for two vaccines, and for sure, because it's adaptable technology, we are working on the commercially available vaccines currently to apply our technology to these vaccines. And we hope that one day we can have this technology available for all vaccine manufacturers to be able to deliver the vaccines all over the world without the need for refrigeration.
Saher, I understand you're at McMaster, but Carlos, you are not. So can you tell us where you're coming to us from?
I'm currently in Portugal, so hopefully returning to Canada in 2 weeks, I hope.
And how do you find that connecting to your research group back at McMaster and now a startup company from Europe?
When you have a person like Sahar, you don't have to worry. Things happen, magic happens. And I think that you can do a lot without actually being physically present.
What do you see as the biggest challenge that still exists in terms of bringing this to market?
I think that the biggest challenge is going to be the translation from a concept that works very well in the lab to the realities of an industrial application. The scales are just so different. And we are extremely good at pipetting small amounts. We are extremely good at evaporating liquids in small amounts. But when you have to prepare millions and millions of dosages consistently, it's an engineering problem, but it's a problem that we know we can solve. It will take persistence and problem-solving skills to get there. Are we there yet? No, we are not. But I think that in the last couple of months, we made some substantial progress in really making this technology applicable to large-scale industrial applications.
One of the things that remains a hope for some of us, certainly for myself, would be the complete eradication of SARS-CoV-2 and the end of COVID-19. In order to do that, that would involve eliminating the virus in the most remote regions of the world, some of the places where it's most difficult to get vaccine to. Is your innovation potentially a solution to that problem? Is there an opportunity perhaps that will one day be rid of COVID-19 by getting that last person vaccinated and eliminating the disease?
Yeah, that's an excellent question. And I think it's been the focus of a lot of work that has been done by the Bill and Melinda Gates Foundation. And they've been investing billions of dollars to eradicate polio. That's been a lot that has been learned by trying to fight that fight. And it's the challenges that are associated with eradicating a disease are a little bit unusual and unpredictable. It goes down to some specific areas of the world that people just don't have a fixed residence and move from one place to the other. It is convincing people that have a certain set of beliefs that vaccination is a good thing. There is so much that technology can do for people, but there are some times that people have to help themselves. I think that's from reading about the polio efforts that have been made, I think that technology is not going to be the solution to fully eradicating any disease.
Okay, I'll ask you both this question. Final thoughts on the impact of this work. If all your dreams come true, what's going to be the outcome from this? Starting with you, Sahar.
Something that really excites me these days is that how important is the work that I'm doing, especially currently that there is a high demand for getting COVID-19 vaccines. I can see better now that how important is the whole process of vaccine development. So we all are waiting for a day that we can have this vaccine available, but if we have the vaccine, there is a high demand also for delivering that vaccines all over the world. So that is the part that I'm doing now. So that really pushes me hard to work harder and to get this technology to all the available vaccine manufacturers faster, because if we have the vaccine like that we know works very well for example, for COVID-19, but still there is a high demand to transport that vaccine in an effective way without the cost of refrigeration, and like in such a way that vaccine maintains inactivity. And that's where my role is. And that's really excites me because I know that my job, something that I'm doing these days in the lab, have a great impact all over the world.
Carlos, do you have anything to add?
My all dreams will come true when a single person would benefit from this and actually would get vaccinated that otherwise would not, and that a life is protected. That will be my dream come true.
Thanks for listening to this episode of Big Ideas for a Changing World. If you enjoyed the show, be sure to leave a review on Apple Podcast or let us know your thoughts on social media. We're at McMaster Engineering on Twitter, Instagram, and Facebook. See you next time.